Now, let's take the standpoint of the bacterium. It's making this huge transition from the free-living bacteria through invasion, in which the bacteria are able to form an infection thread and penetrate through individual cells, and then into multiple layers of the root, and finally nitrogen fixation, where the bacteria have become endosymbionts inside the cytoplasm of a plant cell, and they persist there in a differentiated form, almost like an organelle. All of these changes are accompanied by carefully regulated changes in gene expression. We can take bacteria and through mutagenesis and then blind screens, we can ask what are the most important genes for symbiosis. Now, there's a couple of different symbiotic phenotypes that we have observed. In contrast to a wild-type situation where the bacteria growing in the free-living state are able to interact with the correct host plant and make functional nodules, shown here by having a red color reflecting the leg hemoglobin that's inside the plant. The nodules harbor bacteria that fix nitrogen. The plants are able to use that to make chlorophyll and protein. They flourish and they're nice and green. But if the bacteria have a mutation in a gene that's required for the formation of the nodule, the result will be that the plant forms no nodules, therefore it's not getting any fixed nitrogen, and it starves. It's yellow, and it doesn't grow very well. We would call this a nod gene in the bacterium because the mutations in that gene result in the so-called nod minus, no nodule phenotype. The other outcome, again comparing to the wild type, would be that a bacterium might be able to form a nodule, but those nodules don't function. And what I've shown here is I've symbolized that by these empty white circles. Those show nodules which are not fixing nitrogen. They don't have leg hemoglobin. And so we would call this fix minus phenotype, and the corresponding bacterial gene is going to be a fixed gene. Bacteria tend to have genomes between about 1 million for the very smallest pathogens, all the way up to 10 million bases. Rhizobium is on the large size, over 6.5 million bases, divided into 3 replication units. The chromosome, 3.65 million, and 2 very large so-called megaplasmids. They're called megaplasmids because they're more than 1 million bases each. PSYMB, 1.68 million, and PSYMA, 1.35 million. The nodulation and nitrogen fixation genes are located on PSYMA, but in addition, there are symbiosis genes on PSYMB and on the chromosome. So to some extent, the symbiosis genes are dispersed. Nodulation genes here are the genes that encode enzymes that synthesize nod factor. I talked about nod factor, and I'll show you a cartoon in a moment. It is the substance created by the bacteria. That's a molecular signal that causes the plant to form the nodule, and it also causes transcription and a number of other events in the plant. So, powerful signal molecule. The genes in the bacterium that are responsible for producing nod factor include the common nod genes, nod ABC. The transcription of nod ABC is controlled by a promoter that includes what's shown here in this oblong, the so-called nod box, a highly conserved motif that's in the DNA of upstream of the nodulation genes, both this set of common nod genes and others that are coordinately regulated. All of these genes, as I mentioned before, are located on PSYMA.